Well, yeah, thanks for the introduction, and very happy to go after Mohammed's talk, which I think gave a really nice view of the fast-moving field of reading protein sequence and structure, kind of at large scales, and especially in meaningful ways, like through thinking about protein and molecular interactions. And so now, following that up, maybe I'd love to review some of the motion towards writing proteins, like creating new structures and functions and sequences, and hopefully kind of continue to focus on this bond between AI and science, because I think a lot of the methodologies right now are really bring like a kind of rich set of ideas from physics and other fields to bear. Cool. So as we know, proteins are pretty cool. In fact, for many problems that we face in not just in medicine, but also, let's say, sustainability and material science, they are kind of one of the dominant functional molecular classes. A lot of molecular function on Earth has obviously been created in proteins through evolution. But also when we think about industry today, whether we're talking about like how to treat complex disease, or how we responded to COVID, or building kind of new sustainable materials, the answers often could be proteins. And what's cool is that these little pieces of nanotechnology, unlike, let's say, the kind of extremely advanced semiconductor manufacturing that we all rely on to kind of continue to move ML forward. Let's say, like, if we need to make an H100 on a $200 million extreme ultraviolet lithography machine, kind of once we have the idea of how to build these, it's extremely complicated to manufacture them at scale. But proteins, once you have the idea, making them essentially requires the same technology as making beer or kombucha. So one interesting thing. So now to kind of follow up, we saw how there's this tremendous protein universe that we're finally being able to kind of grok at the level of sequence structure and function through awesome work like with modern structure prediction methods. But when you look at industrially relevant proteins, I would say that kind of most of the things that really matter are often taking what nature has already created and editing it. So for example, if yesterday when Yisong talked about kind of some of the important work in collaboration with Arnold Lab on evolution of enzymes, like one of the great workhorse enzymes is kind of this family called the P450s, and they can do a tremendous amount of chemistry, and kind of that's making some of the best catalysts that we have has been about taking things that already exist and kind of editing them in some ways. Similarly, stuff we do. I'm at this company, Generate Biomedicines. We make new proteins, but a lot of the ones that like near term we're going to put in people will be antibodies for a lot of reasons I could explain more of. But like this is also taking essentially what evolution also found, which is that kind of building a class that can be easily edited is easier than kind of inventing things from scratch. And I think just stepping back from this for a second, it's interesting to ask, why are we so entrenched in terms of kind of evolutionary function on Earth? And maybe first, if you'll excuse an aside, but this is like AI for science, so let's like put on phylogenomics hats. Like it's actually really interesting where just all this tremendous protein diversity on Earth came from and when it came from. So this was a work from Eric Alm's group a bunch of years ago at MIT. They can do like phylogenomics. This like takes just all the sequences that are around and kind of models of evolution to create best guesses about like when certain gene families arose and kind of did they get passed between organisms and genomes versus kind of get edited or duplicated from other things. And just the main, like without kind of going through all this detail, first thing you notice, this axis is billions of years. So, or talking like up to 3.5, 4 billion years ago. Red is when protein families were born. So what you see is kind of most protein families got invented almost 4 billion years ago, and the name of the game since then has been copy, paste, and edit. And of course, like one like very reasonable argument is just like the biochemical repertoire on Earth kind of got a useful set of domains and functions that kind of actually already could host most of the molecular functions we need. And like while protein space may contain many more, actually you can kind of actually just get a lot done by working within that kind of vocabulary that already exists. But it's interesting to ask like why why is this so hard? Like, why is it easier to kind of keep editing things that already exist? Then sure, go ahead.
When you see the changing ratios between different colors, like, did the condition on Earth change to cause these changes?
Oh yeah, that's like kind of one of the headline points of this paper in the figure. So they talk about this as like an Archean genetic expansion. The Archean this is like even Precambrian in a sense. And one hypothesis is that, especially when you look at the specific types of biochemistry that was evolving, it was like related to an increasingly oxygenating atmosphere, which allows you to do like more redox chemistry. And so like a lot of the things associated with that seem to kind of follow suit. So yeah, there's, I think, probably a lot that potentially speaks to the changing both like Earth environment and also kind of ecosystem, like what organisms were around. But yeah, that can go become its own big deep dive. So as we saw earlier, and this is kind of me, I know this is a question in the last talk. I think we had like one local news coverage like talk about protein diagrams and called them confetti diagrams. But like when we show these diagrams that have kind of emphasize the secondary structure, really what those are kind of summarizing or hiding is that there's a very complex, relatively like small length scale constraints that need to, in some sense, be exactly satisfied to kind of be a realizable, foldable, happy structure. So obviously proteins, they're polymers. They have to self-avoid. There's, the dominant force potentially is like hydrophobic. Put the things that dislike water on the inside, the things that like water on the outside, but then kind of do that also in these kind of confined, hyper-specific geometries that involve these hydrogen bonding networks that the backbone polypeptide likes to make, independent of sequence. So all the kind of alpha sheets and beta strands, that has to do with what the backbone's doing, not the sequence. But then we've got to kind of reverse engineer things that will satisfy all these constraints while being realizable as sequence and have a pathway to fold. So it's hard to bump into these constraints by accident. But then like if I start telling a story like, oh, proteins are so hard, they're so complicated, I feel like in the era of modern generative models can sound a little silly. I mean, it's not like images don't have a lot of very complex multi-length scale constraints, like down to, like just the amount of pixel level constraints required to implement complex patterns, let alone like higher order like constraints to satisfy semantics. So I don't want to just be like kind of biochemist person making excuses. More the question is like, how can we start to see some of these benefits in the world of proteins? And especially if the problem is that it's kind of hard to land on a protein that will satisfy all these constraints at once, can we kind of accelerate this process with like modern generative modeling? So the classic solution to doing this, and by classic, this is really kind of like the 2000 through late 2010s, but the ideas went back to the 80s, is really one where we're going to try to satisfy these complex constraints via deeply iterative search methods and kind of attempts at describing all the constraints at like very atomistic low-level energy functions. So when we're talking about designing de novo proteins from scratch, this is all like largely coming from work out of David Baker's and the kind of Rosetta extended community, but we kind of think about two things, both like how do I make a sequence given a backbone, how do I kind of design a backbone where to even evaluate it I need to also instantiate a sequence with it if I'm going to criticize it with these low-level atomistic energy functions. And so it's just lots and lots of deep searching, and at the same time we didn't have kind of even, let's say, great critic functions like we heard about before, such that even if you could create something which searches this process, it was hard to know what's going to work or not. And then, in the past few years, though, this is probably, so we just heard about kind of how deep learning has been kind of taken over protein modeling and understanding protein molecular modeling. It's also taken over de novo protein design, designing entirely new things from scratch. So both the problem of kind of how do I make sequences given structures, this has kind of become like many groups that were designing proteins from scratch. It's just now it's faster, it's cheaper, it's more robust and reliable to use neural networks to design sequences given structures. And then there's also just been this awesome flurry of research to kind of also start to see, well, can we use ideas like diffusion models to design everything from scratch? And I think it'd probably be fair to say that like deep generative modeling has essentially eaten this field now. I think the last 3 of these papers, one of which from Mohammed's group, even just in the past like few weeks. So it's a really exciting time, lots of cool work coming out. And so what I'd like to tell you about today is something we were involved with. This is just one of these papers. You know, there's a lot out here over here. And some of the lessons that we learned. So we kind of make models that make proteins that we make in the lab. And we kind of learned some things in that process of trying to do it. So maybe I can share them with you today. Second time the diffusing dog has been presented at this conference after a Swan's talk yesterday. So it's about like making hard, complex constraint satisfaction. Diffusion models are this like really powerful paradigm where we kind of reduce that into something that can be learned incrementally via regression. Just rehashing, we want to define a forwards process that destroys structure in data. Learn an optimal where did I come from estimator. So this is like generally like a posterior estimator of the posterior mean of where did I come from to arrive at a particular noisy sample along the process. And then it turns out you can parameterize the kind of smooth time-dependent distribution in terms of an optimal denoiser. And also there's a bunch of other related frameworks like flow matching, score matching, other things. They're all just like affine transforms of the posterior optimal denoiser. So in some sense, they're all kind of learning the same things and sort of with kind of different preconditioning. And since this is the AI for Science conference, and we're like trying to think about this, and I know Stefan mentioned yesterday that it's really cool to see how, like, of course, if you look at the original work on diffusion models, it came from like non-equilibrium thermodynamics, Derzynsky inequality stuff, and kind of Yasha and Surya et al. It's also quite interesting that I haven't seen anyone point this out yet, that the protein modeling world was doing this stuff in the 80s. Sharaga was this professor who did a lot of early work on kind of protein folding, the driving forces of protein folding, molecular modeling, coarse graining, many other things. And they actually were doing this thing all through the 90s, started with this paper, I think, in '89, where they were basically like, we'll take potential energy functions, we'll convolve them over Cartesian space with like Gaussian kernels, and then we'll, seeing diagrams like these in diffusion world. And then we can do the kind of turn the like rough, rugged landscape sampling problem, we can like do like very gradual walk back and kind of trace back global minima kind of along these basins in a way that is like extremely evocative of kind of modern diffusion stuff. No, that's a really good, okay, so this is a very good question. I know a lot of times in diffusion modeling, we talk about temperature, and kind of smoothing is like temperature. But smoothing does one thing that kind of classical temperature does not, which is it puts non-local information in the local gradient. So essentially, if I do parallel tempering, which is a temperature parameter, and I'm like at high temperature, my dynamics devolve into a random walk. Whereas if I have one of these smooth distributions, the locations of basins at the smoother kind of perturbed potentials are informative and will guide me to things on the kind of finer potentials. So I think that's kind of parallel tempering was like almost there, but it doesn't kind of the same thing where like why we can't train neural networks without gradient-based methods. It's because we need that high dimensional signal of like where do I move in high dimensional space. Yeah.
Maybe this is off track, but doesn't that require, if we were to apply this to modern diffusion models, either training multiple reverse diffusion processes, because in order to encapsulate global information, you have to like build a new model, a coarser model.
Oh, I would say like this is baked into modern diffusion models. Learn like the T parameter indexes this family of distributions.
I see. So in this diagram, it only goes in one direction.
Right. Well, so the cool thing, if you like go,
it doesn't jump back up.
Yeah. So they did a bunch of stuff. Actually, there were like a bunch of papers all through the 90s. They actually worked at this cool thing because like diffusion kernels are linear, which means they apply to components of energy functions distributively. So they could actually work out by hand like in a molecular potential, what the effect of diffusion kernel is on them, and like work out like a kind of analytic approximation of like a time-dependent potential. So it was like a lot of that kind of thing. Now we have the awesome ability to just bake these smooth distributions into neural networks with regression. But I just, in the spirit of kind of pointing out things, I haven't seen folks kind of point this out yet, and it was like cute to notice it recently. Cool. Okay. So what can we learn building generative models for proteins kind of in this modern paradigm? I want to talk about 3 things. And the first of which is just kind of something that maybe so we just heard a lot about how like the most successful structure prediction methods have in large part relied on kind of cubic or quadratic architectures, and it's like this is intuitive for like really well argumented reasons. So I have to consider all possible interactions in a molecular system if I'm trying to predict over the space of all structures, like what it will adapt. Or if I'm like thinking about embedding Cartesian coordinates in 3D space, like I want to think about kind of metric relationships, distance geometry, et cetera, maybe where I get like cubic reasoning. At the same time, if we look at like things, properties of networks that have scaled, like image diffusion models, most at least like kind of convolutional neural network-based approaches, like they scale with the number of pixels. So linearly with kind of the dimensionality of the data, not like super linearly. Then you can say, oh, well, proteins need interactions. What about long-range context? I just want to like, this is so here, I mean, you can basically always do better with more interactions. What we were super interested in was like, can we scale these models up to kind of meet large complex systems, train on them at their full size without cropping? And like, can we get away with less? And like one motivation for why this should be true is that like if you look at kind of what have been the computational recipes in molecular simulation in physics, like usually eventually someone comes up with like an n log n algorithm that takes advantage of the fact that there's kind of smoothing or hierarchical structure in physical systems and interactions. All the way from how you do gravitational simulations down to kind of stuff that's used in molecular dynamics. We're not doing this in like particularly fancy way, but we can like think about properties that are important that like I want to pay attention to far away things, but I don't want to kind of do so at the expense of preserving local attachment. And I also want it to kind of scale gracefully with size, such that kind of the implied marginal distribution of distances is monotonically decreasing, as opposed to have some like weird stereotyped, like our most typical connection is like a certain number of angstroms away. And when you do all these things, you kind of end up that like there's a certain type of random attachment model that produces these scaling behaviors. So interestingly, this ends up also being the scaling behavior that to kind of make it continue to scale without like stealing local context in large systems. This is an n log n method. It was inspired by essentially the like scaling of Barnes Hut methods. But I mean, honestly, that was kind of just trying heuristic analyzing these things. I think this is also reminiscent in like transformers. This whole kind of idea is a little bit questionable because like right now there's also a question of even big transformers like has subquadratic attention ever worked so well, or do you just get better hardware implementations, scale up ring attention, do stuff like that? But there have been ideas like Big Bird or others where you kind of have some guaranteed local context and then dope in long-range interactions. So we're doing something like that. We're thinking, well, if we have this, yeah, go for it.
That was not the most provocative, I think, part of the Coma paper.
Sure.
Yeah, I mean, it's great. But have you done the kind of the ablation, whatever, where you've tried a denser? Like, do you know how well you would do versus the dense network?
We don't have anything public on kind of, yeah. I mean, I think you can definitely do better with dense. Yeah, I'll say that. And we're like we're definitely not. This was more like existence proof for us. I'm like definitely not against dense. Especially kind of sometimes maybe dense is easier to implement on hardware. We did at least see we did some ablations on like how you use your edge budget. And it seems like it's good to do this like a little bit of long range and if you're forced to be kind of cheap. Okay, so then we're also thinking though, if we're in this regime where, well, at least when we were trying this originally, kind of a lot of the network was spent like relearning kind of simple chain constraints or kind of other properties of proteins. And when we have this sparse architecture that has a strong inductive bias for spatial locality, like that may then be especially sensitive to the choice of diffusion process. That kind of is destroying structure. And one thing we found is actually it's like not hard or cheap or even mathematically complicated to build covariance structure into the diffusion processes, especially if you just have like a flat covariance throughout what's going on. And you can actually, there's like simple toy models that have come from like the biophysics literature of actually when people were like analyzing hydrophobic burial and collapse. Yeah.
When you say distance preserving, does this mean that you parameterize sort of the angles as opposed to with bond lengths, or? So
it's actually it ends up being like a prior that we build directly a Gaussian on Cartesian coordinates, not like a nonlinear reparameterization or internal coordinates or something. I think internal coordinates are still hard because of like lever effects. But essentially, if you just have this, so this is our covariance model. It's really simple. This is the inverse covariance. So it's essentially like you just have chain springs along a linear polymer. And then they also have global springs that pull you to the origin. And you just set the parameters by essentially like fitting well-known scaling relationships in proteins from biophysics. So basically, there's like a certain relationship between like the radius of gyration of a protein and its length. And you can kind of just set these parameters to match those. And the nice thing is because it's so simple, you can work with this in linear time. And it basically kind of doesn't really cost anything to kind of work into the pipeline. So putting these together, we're interested in the existence proof. We also do some other stuff about kind of how you parameterize equivariance. As we keep talking about equivariance, is it necessary or not? I mean, this was kind of just something that suits the architecture well in how it was designed and with the sparse architecture. We'll be continue to be interested of like what are the best ways to parameterize, or should this just be kind of Cartesian input-output non-equivariant? But it's something that works well with these kind of sparse graph formulation. We also, it will come to this later in the talk, but kind of using essentially the same architecture, like a graph neural network, just operating on these protein graphs. We also like predict sequence and side chains off of that, so you can kind of go back and forth between these two things. I'll come back to that in a bit, but punchline, existence proof, well, we can at least check the box. This can at least do stuff quickly. So we're using, it only uses like 60 edges per node, and so we are able to train. I think like even AlphaFold 3, it doesn't train on full complexes, but on kind of subsystem crops. But we can like train on up to 4,000 residue systems, could be larger if we wanted. I'll just go again. But yeah, no, we can do the diffusion thing and kind of meeting complex data where they are with these kind of minimalist, just GNN style architectures. Yeah.
Can you say a little bit about what the prior is in the diffusion? Like, what are you diffusing to? Is it polymer chain?
So the prior, it's actually so I didn't actually end up saying this here. So our prior is, it's just like we're going to a Gaussian, except instead of the Gaussian being like a standard normal, we are converging to this essential, what you could call like a quadratic model for like random Gaussian globs. So it's essentially satisfying the statistics. It's essentially like a random coil, but that satisfies that the residues are close to each other, the chain length is preserved, and then these like there's no like control, like as soon as you have like real constraints on steric overlap, that's like nonlinear, and it would be cool, but it would kind of like break the whole beauty of diffusion regression training. But this we can just like it turns out that kind of just by statistical coincidence you can get approximate occlusion simply via like matching empirical RG scaling. So it's basically kind of just the dispersion comes from randomness, and there's like an effective, the effective space filling is set by kind of how tightly all the residues are being pulled to the origin or the center of mass. So actually Jeremy England's lab had like some work where they even showed you can like in this type of Gaussian spring setup, if you like customize these springs to like match hydrophobicity, you can like maybe predict some allosteric kind of properties of proteins in very specific regimes. Okay, so we can do the diffusion thing. There's lots of cool diffusion models going on. So we get asked to make proteins a lot. And I think one thing that we found, just to kind of organize this effort, is like the constant question of like, every time we have a new, interesting type of protein we want to make that has some kind of complex set of constraints to meet, like, let's say, some therapeutic idea we kind of have to, do we train a new model? Do we have to build a new infrastructure? Do we have to do this thing? Or kind of can we inherit what used to be true for protein design, which is kind of you have these energy functions that you can kind of mix and match with your favorite kind of sampling constraints and algorithms. So can we kind of make it more compatible? And it turns out there's like lots of types of conditioning and constraints you can want to do. Like one simple version of it is like soft constraints. Like I just want to penalize it so some property is kind of softly satisfied, like a score is kept low, or you can think of that as like a likelihood term. The nice thing about like score functions is that they are additive because they like the score function doesn't depend on the normalizing constant. So like the posterior score function, if I have this for my prior, if I have some guidance term that's time dependent for my likelihood, I can just add them together and I can do Bayesian inference in score space, and I don't have to worry about normalizing. However, like sometimes we also want to do hard constraints. It's going to be a little trickier to think about, and also things that might not even fit necessarily into kind of the posterior inference setup. Okay, I'm gonna after Stefan gave like a lot of nice talk about kind of all the like parameters when thinking about diffusions, samplers, and processes yesterday, I just want to make one point here. So as we know, like the name of the game in diffusion modeling is we're turning kind of our data distribution into a simpler distribution through the sequence of smoothed intermediates. And there's a bunch of ways to formulate it. One of them is the SDE formulation. And there's kind of also this well-known, if you look inside of the reverse SDE, there's actually two things going on, kind of infinitesimally per time step. So we can do this awesome, like parameterize the reverse the error of time, but if we know the score. But then inside of this expression, if I just like split this term in half, I have two things going on. One is essentially a purely non-equilibrium process that takes me between marginals. So kind of no mixing within a distribution and just kind of walks me through the level sets of the marginals in this high dimensional space. That's the probability flow ODE. And then I have another process which is purely mixing within the current marginals, purely equilibration. And so if we're talking about perturbing this process, the annoying thing about diffusions is that like the non-equilibrium part is only valid when we kind of trained or kind of have made special considerations to kind of make it fit the like particular forwards process. However, if we want to, let's say, add other constraints that will kind of add in a time-dependent way or kind of from just some other source kind of into this diffusion process, we kind of will be, it won't work out of the box. And so, but the nice thing is that there's like rescue for this built into the reverse SDE itself, which is just that like, if we, for any sequence of distributions we might construct between kind of the prior and our data distribution, and hypothetically, enough sampling can kind of catch up to the perturbation we make, in the limit of kind of sampling and things not getting trapped in mode. So if we kind of do something, if we perturb these in a gradual way, it no longer fits the non-equilibrium picture, but we can just upweight the amount of equilibration per time and kind of get around this. Okay, so this is like essentially the idea of how we're going to try to do lots of different conditioning things in the same framework. So first off, I mentioned, so the awesome thing about diffusions is we just learn a denoiser, like a posterior optimal, posterior mean estimator, like what is x naught given my current xt in a time-dependent way. And then like, while we don't always explicitly write it out, you can, like that is technically the score, that's a gradient function, is the score of some potential, which is looks like this, which is like a Gaussian likelihood. This R is just kind of our correlation stuff that we have going on. And so now we're going to just kind of do the standard MCMC thing. So when you do MCMC and you want to add constraints, you reparameterize your system. Or if you want to kind of add perturbations, you just add restraints to your global energy function. So if we start with the base diffusion energy expressed in terms of the optimal denoiser, we can add in coordinate transformations. So we're going to like rewrite our time evolving state as like kind of the transform of some unconstrained coordinates. So x tilde is now like the state variable, and we push it through some transform that's, let's say, like enforces a certain property, for example, symmetry. And then we can just add in, like in more the traditional Bayesian likelihood way, kind of a restraint term that's kind of tilting this distribution to enforce whatever constraints we want. For example, classifier guidance. So then now, as you said here, as long as we just upweight the amount of Langevin that's happening per time, but you do have to do this, we can basically screw up the energy and kind of surf it as we go along. So as long as we're willing to pay, so this is not free, you know, it's still a great idea if you have conditioning that you're always going to want to do to train your diffusion model to like respect that conditioning. But if you kind of also have this long tail of random things you'll need to do sometime, kind of that picture is not incompatible with like basically messing with the energy. And there was actually some really nice work from Will Grathwall and Yulin Du on kind of this type of thinking of like if we are free ourselves from kind of trying to be non-equilibrium all the time, and we get more equilibrium, and we just use MCMC to traverse the diffusion marginals, we can be way more creative. We can do and functions or functions in diffusion conditioning, things that completely break the non-equilibrium picture. But then you just do more Langevin sampling or HMC or something. And this is also the hard constraint version. This is kind of related to if you've seen like Dream Fusion, how folks kind of parameterize, let's say, like have a 2D image generative model explain an underlying nerf.
Yeah, super interesting. Could you explain again why you would actually want this sort of mixing between the marginals? Because oftentimes it's not really needed, right? When you just do conditional or unconditional generation, you kind of just you basically just simulate the deterministic equations and you don't care about the stochastic part. So in your application, why do you care about it?
Yes, totally. Okay, so maybe if I get, I don't like my next. So basically, if you don't mess with the energy, it's a waste of time. So you don't want to do it. However, if you make certain changes to the energy that violate the fact that your score function is the correct score function to kind of reverse the correct conditional process, then it will break. Actually, one of the simplest examples of this is temperature. So I think this is like why people found temperature sampling with diffusion models kind of hard. Because if you just naively take the score and upweight it in a kind of temperature scaling kind of way, it often would screw things up a lot. And Will explains this well, or they explained it well in this paper here. But that's essentially because just upweighting, the temperature upweighted score function is not the correct time reversing thing for kind of for the process you're implying by that. Does that make sense? Well, yeah, and maybe it will also be clearer in the applications. Yeah, we definitely can chat offline.
Can you explain how you're using equilibrium? You mean like there's a potential, but you're changing it in time, right? So I mean, everything that you just wrote depends on time.
That's right. So if you kind of take a continuous picture and imagine that we're doing, for each chunk of time, we're doing some amount of transitioning between distributions and some amount of equilibrating to the current distribution, I'm saying we need to do more of the latter per unit time. You could kind of make an extreme version of this, and actually Song et al. Did that in the original like SDE diffusion paper with this kind of predictor-corrector approach, where it's essentially kind of like predict, transition between the marginals, and now kind of get back on the manifold by equilibrating for a bit. But like you can also just, for implementation's sake, you can also just write down an SDE that has these two notions, and if you kind of just put a bigger coefficient in front of the diffusion, you're essentially equilibrating more for time. Yeah. But now this is like the kind of annoying, like formalizing it. Really what you want to see is in action, how we kind of use, like what am I talking about with these Fs and Us. So this was kind of we tried to take all the kind of protein things we'd imagine we want to do and smush them in here. So first off, I can just say at the highest level, you can imagine, for example, some constraints and restraints. So like in this setup here, if I want to add a likelihood restraint, like classifier guidance, that's just going to be adding, P of y given x, like a gradient to the energy update. If I want to add a linear constraint, like symmetrization or kind of substructure, something that like enforces kind of a linear projection on the system, then that's actually just a state transform of the system. And then nonlinear constraints you can do with like the change of variables formula, where I have like some function that like maps my unconstrained space to a specific manifold I care about, and then as long as I correct for kind of the change of volume term, that's good. And you can like fit a bunch of things in this picture, and I'll tell you about a few of them now. Cool. Okay, right. So part of this was also just, can we build like sustainable ecosystems that we kind of can build up around? And the nice thing is, because this idea of these state and energy transforms they have the same input and output type, we can compose them with each other. So, so I can, for example, think of these things as blocks. When I want to plug this in, I can write one sampling algorithm once, or I can, like for my various sampler algorithms, implement them once. And then for any conditioner, it's essentially like a program that takes like an unconstrained representation of the state. We push it through the conditioner, and this is very much, just like parameterized differentiable functions. So this is like very PyTorchy or Jaxy or your favorite frameworky. And then tack on the global energy computed on the final state at the end. And if I backpropagate through this whole thing, it gives me like the correct gradient to do Langevin on. And then I can kind of compose them in some regimes to kind of express more complex conditioning constraints. So without like kind of going into implementation details, it's like just as we just were like proof of concept. Let's say I want to say my constraint is that I want to make a lattice. So my hard constraint is like that's a linear constraint, because I essentially take one unconstrained unit cell of the system and I have like a tessellation operator that just copy pastes it a bunch of times. I put it in this framework and then plug it into a general sampler, pulls the gradients back through, and it kind of, just by virtue of the chain rule, automatically does the kind of, symmetry conditions in the gradient propagation, and you can automatically, sample a lattice system. Okay, so we do this for a bunch. So now I'll show you kind of like a gallery of conditioning. So symmetry is a simple, that's a simple state transform. It's basically like I have an unconstrained representation of the unit cell, and then I have a matrix or a tensor that kind of applies all the rotations in a given symmetry group to that, backpropagate through the whole thing, and get symmetry. It scales up to, since we have our nice, cheap scalable architecture. This was interesting. Substructure infilling. One nice property about our correlated diffusion prior that preserves the chain constraint is it actually already has somewhat interesting kind of guardrails that it could hypothetically apply when you think of it as a conditional prior as well. So it's essentially technically like an OU process along the backbone, or like kind of like a Brownian motion with a little restoring force along the backbone. And so then if I wanted to say, I want to create now a conditional prior for like how a chain satisfying thing, like if I want to take the Gaussian that's kind of fully unconstrained and I clamp two parts of it and I want to know how the intermediate can move, that's actually something you can do analytically, and you get these kind of like Brownian bridge or OU bridge type priors. And the nice thing is it's literally, this one to implement was like 3 or 4 lines, because we basically just need to undo our original covariance matrix and like reapply the thing that brings it into our other covariance matrix, backpropagate, and now we're kind of doing this Brownian bridge motion, and it exactly satisfies the constraint by construction. You can do, so we did, here we're doing optimal transport. So this was something kind of the DNA nanotechnology world had done this a long time ago, and I always felt like the protein, protein's got to catch up someday. They're smaller. Knowing the difference between nucleic acids and polypeptides is good. They're like, these are actually like quite smaller than kind of DNA nanotechnology. But basically this was just simple OT. So if you have like a point cloud specifying your shape of interest, we can do like optimal transport, like a Wasserstein distance from kind of the current atoms to your favorite like target shape of interest. We kind of had to do this one extra bit of regularization that like tries to make the chain fill it more ideally. But it again, it's all we're using the same sampler for all of these, and we're just changing these kind of state and energy functions as we go along. And then this was super early, also more of an existence proof, where we're just asking about semantics. So I think we all are seeing like the benefits of natural language interfaces, but if you had something that could predict properties from sequence and structure or structure, or even text annotations, can we like invert that in a classifier guidance way to start sampling like functional protein function on demand? And in that case, again, it's just like adding classifier guidance, that kind of sense. So I have lots of things. There's lots of types of interesting applications you want to do with these, like symmetry. There's a lot of interest in nanomaterials, nanoparticles, for hosting kind of agonists or things, vaccines, delivery systems, substructures related to some things around designing de novo binders, kind of grafting bits of enzymes. Didn't go into like all of the conditioners there, but again, it's kind of like write your own ecosystem. Yeah.
Constraints throughout the entire diffusion process, or only at the end output?
Throughout the whole thing. And in fact, like I think that's kind of what makes it all work, is that it kind of breaks apart the complex satisfaction process of like make a thing that's a protein and satisfy this kind of potentially complex constraint into this like sequence of subproblems.
Whether I satisfy them later or earlier on in the process, or is it all static?
That's definitely like a kind of hyperparameter. You can like put schedules and these kinds of things in it. But unfortunately, it's still often that. It's like requires some tuning.
My second question is, are all of these, do you have any cases where the process is not differentiable with respect to x, like p of y given x is not differentiable with respect to x? Or are all the cases you can possibly think of, you have this differentiable operator?
You absolutely can, actually. So one thing is in the state transforms, like you often, you can do like stop grad things and kind of fake it. So yeah, I guess technically most of the time we're using differentiable transform. There may be workarounds, but yeah, no, for the most part, yes. Okay, but so was that just a bunch of pretty pictures, or is there any plausibility here to that? Like these are kind of relevant and things that can be useful. So I want to talk about like in silico validation and in the lab validation. I mean, I think the punchline is, unfortunately, we as a field, we're and others, we're all trying to go ahead.
I'm curious, you apply these like symmetry like constraints. What if you start, instead of starting with noise, you start with an already existing protein and it's trying to diffuse from there? Like, what happens in that case? Are these like, do existing like proteins, like our structures already, like, do they already fully satisfy these constraints, and therefore like the gradients are kind of zero in those directions? Like, what happens? Does that provide any additional like information?
Yeah. So if the question's about like, can we put like hard constraints on things that don't satisfy them? Well, the very nature of them being hard constraints is that like from the beginning to the end of the simulation, it's exactly satisfied. You can either kind of work out initializations that try to like squeeze something that doesn't satisfy it to project them into something that satisfies it, or you can turn like constraints more into soft restraints.
I was actually asking not so much like if you start with a protein that doesn't satisfy, but like take a protein that already satisfies the constraints. Like, what happens if you do diffusion? I'm just kind of curious, like do these constraints?
If you're not doing de novo.
Right. Oh yeah, so you can also do partial diffusions, and it's a very useful relaxation operator. And I think like us and others have, like that's already, doing partial diffusions is also a very useful thing to do to kind of locally diversify or re-optimize kind of around structures that you already have. And you can also do that in the kind of constraint-based setting. Okay, so then we have to, I'll talk a little bit about the in silico part and then make sure we get to the lab part and with time for questions. So first off, I'll say with the in silico, it's like still very early days. As a field, we're all trying to figure out what are good metrics and how do we predict kind of what's going to work well in the lab. And the solo seq method, where that will be cool to see if we can try it at some point. How we're doing this though is kind of, I said, so we're diffusing the backbones, and then you can either do the sequence at the same time or do it at the end. It's often just more convenient to do it at the end. And the reason you can do it either way is because we're technically defining a time-dependent joint likelihood, because we predict the T0 sequence at every step in time along this diffusion process. And this kind of gives us this flexible model. We also had found at various points in time, it's like easy, it can be helpful to like model your kind of conditional sequence given structure with different types of factorization. So we kind of have a choose your own adventure version of like autoregressive, more like conditional language model, or a conditional POTS model, or just first order marginals. Like I just want to say two things about this that were kind of interesting to us, a bit of a surprise. First, some interesting things happen. Like you can actually, this is more just if you have like looked at sequence constraints before and like wonder where do bits of information kind of that constrain a sequence come from. If you look at a lot of multiple sequence alignments and statistics around them, it's a question that sometimes comes up. Well, so we can actually kind of look where sequence is arising along the diffusion process, like how backbone decisions are kind of like implying certain sequence decisions. And it's, at least in ours, kind of along this process, what we see is kind of now at the T0, you know, there's lots of like hyper-specific kind of very geometry backbone dependent, like this has got to be a threonine, this has got to be a proline, this has got to be a glycine. And actually as we go further out, let's say to like this, like halfway through diffusion, ours is kind of like slow noise ramp up because it follows the, one of the schedules is like better for likelihood performance. But so with the halfway through the diffusion, which is still like you see pretty modest, just like a few angstroms of a noise bubble kind of around the initial thing, we still preserve like a lot of the structure, but a lot of the kind of exact sequence precise things like that I want to shove some alanines in here, or kind of very like specific surface choices like devolve back into like, okay, no, this part's hydrophobic, this part's hydrophilic, maybe some opinions on kind of the precise hydrophobic packing that will solve it. And then this was another, we also, because we have a conditional POTS model, we can look at some of the POTS couplings that are in this time-dependent way in the diffusion. And one kind of, this is now extremely anecdotal, but at risk of saying what we see is also that when you kind of go to this slightly noise-robust part of the diffusion, then this is now anecdotal, kind of have to take my word for it because I like stared at a lot of POTS model coupling matrices for a period of my life, that like you kind of start to see something that looks a lot more like what you see in natural evolution, kind of biochemical block consistency. So you kind of see like, oh, positive goes with negative, oh, polar goes with charged, oh, certain aliphatic size packing patterns. Whereas kind of it's like at time zero, it's like this super rugged kind of complicated thing. Why am I like wasting all the time on it? Well, one interesting thing is, so also we can look at when you do a refolding test. So you take these, you take the design a new backbone, you put a sequence on it, and now you refold it with kind of your favorite method. And what we basically see is, interestingly, well, we can get refolding across lots of different categories, first off. So even up to the big length 1,000 things, we can get lots of things that will kind of refold even at like pretty tight matches. I know like there's a lot of focus right now on RMSD less than 2, which I think makes a lot of sense in like small molecule active site stuff, but like the breathing of a 1,000 amino acid thing is, I feel like that's maybe like a little tight. We kind of see it across secondary structure distributions, but interestingly, and this may just say that there's something funky with our backbones at low times, but we also like having kind of a robust thing that can like fix our errors just because we try to do spicier protein designs a lot. That robust one, the kind of the T.5 ensemble sequence design bubble, just like massively improves refolding. And if you look at all of them, they end up often undergoing like, you know, just like a 2 to 3 angstrom relaxation. That's like, it's still exactly the topology. It looks great, but it's kind of like or at least for us, we're like trying to just make proteins that work. Having this fudge factor has been really interesting. But all the results I'm about to show you are actually just using the exact design model. So yeah.
I got a bit disoriented. Like you were saying, you can do the inverse folding at the very end, or in your case, you can do it as you're diffusing.
Sorry, that was confusing. I'm doing it at the very end, but I'm using the interpretation of a structure that's induced at the middle of the. So because
to do it at the end, after the fact, like you run the whole diffusion and then you go back and index and do like a midpoint.
Right.
To do the inverse folding.
That's right. We essentially say, well, one benefit is like our diffusion is like protein statistics. So it's kind of going from like this specific fold to like random glob. And now if we take the sequence design network to like pretend it's in the middle of the diffusion, that, at least right now, seems to...
That works better
than using the T0.
Oh, okay, I see. I thought you were somehow doing
Yeah, you can. I mean, the main thing we just find is like a lot of extra computation.
And it doesn't buy anything.
Or at least not in our hands,
okay,
yeah.
Okay, I see.
Yeah, it's still...
Actually, the midpoint is what gives you the best. The T equal 0.5.
Right.
Precisely, okay.
And again, like our midpoint is like a small amount of noise, just kind of because of our scale.
Okay.
Sorry if you said this, but for the sequence design network, is this being retrained on like T equals 0.5 diffuse structures, or it's thinking that the T equals 0.5 is just another example of a backbone, like as if it were trained on the PDB?
It's one network that is told what T is, and it's seen everything in the diffusion. And so then we just show it something that's technically, a T0, but we tell that T is 5.5. So it's like a robustification. I mean, protein MPNN also does like noise robustification. It's kind of like we get a choosier amount of noise just built in by kind of training. And weirdly, we checked, but that like 0.5 is pretty optimal. It was one of those like funny numbers lineup things. Okay, well, so you can extend this refolding to lots of things. We got ESM fold to refold the alphabet, like fun things you can do. And you can kind of across all the conditioners, we could see refolding of these different constraints. But then, like one last kind of the important thing. So we're like, in fact, I think there's more experimentalist authors on this paper than computationalist. We also kind of wanted to just ask a question of what if this is just a proposal mechanism? If we're trying to go out into protein space, like the most useful things are the weird things. So what if we just take raw model outputs, we don't filter them with refolding or anything, like maybe, well, we have to do some amount of like likelihood filtering, even and temperature, like, even GPT needs like temperature. But like, so they look kind of weird. We'll take raw outputs and then kind of prune them through some kind of semi-high throughput screen for solubility and expression, and then kind of go on characterizing them. So we did this kind of with this like GFP assay that like is based on the idea that if kind of things don't fold well, like GFP will aggregate and kind of get recycled or not fold correctly. And we had some kind of controls that would suggest that this does in fact kind of connect to things being well folded. We took a bunch of them and across different secondary structures saw that they gave like nice kind of circular dichromism, which is this biophysical assay that kind of is a readout of roughly what this material's secondary structure content is. So like beta will look one way, alpha will look another way. This is kind of coarse characterization, but it's like a strong evidence, like kind of we have something that's expressing well and it has these kind of nice properties. But then we also were able, of the ones that were on the tail end of the GFP, a lot of them were like super expressors. We got a few that like crystallized super quickly. And even though, interestingly, we're like a cryo-EM company, like we have a bunch of cryo-EMs, they crystallized so well that like that was the fastest path to like getting a structure. And so we got these two things. One fun fact I'll say, is that the first structure we got, if we had done structure refolding filtering, would not have passed. So at least like with AlphaFold does not refold this thing, or AlphaFold 2 at the time doesn't refold it. I think ESM fold does, so it's more like, I'm not saying like you couldn't have predicted it, but I think it's just more like kind of a little bit of this like, we should try the spicy things and see what happens. They kind of don't have hits to nature, but then they still, these were with exact sequence design, like had like quite atoms on atoms kind of reconstruction. And fun thing, so this is maybe kind of future work. I think some of the things we saw today of like building calibrated kind of energy functions, critics, and like the single sequence distillation, super interesting for this. But at least in our hands, we have like all these metrics. Like our model has some likelihoods. All the different folding methods have some kind of TM scores or confidences. We also look at novelty. They all correlate with sequence length really well. So like the refold, it's hard to refold big things. Likelihoods have some certain pattern for like certain size things. But when we corrected for length, like really nothing was particularly predictive of kind of what, at least in our GFP expression assay, kind of what ideas are good. So this is both kind of a challenge, like we've all got to kind of find out what the good metrics are to kind of predict what's going to work well in the lab. But it's also like an invitation that it's like a great time for us all to go and make more proteins, because like these data can help us start to pin down and like better predict. Or like this also might just mean that we're at the edge of what our models are currently good at reasoning about.
Just FYI, there's 3 minutes total left.
Yes. Cool. Yeah. So I'll say thanks to a bunch of people who did it, and then yeah, I can do questions.
Really quick, great talk. Quick question. You said more proteins better. Do you know which type would be the most informative?
To experimentally characterize?
Yeah, like if you had to choose one, it would be the most information gain.
Well, the good news is that today, like, you can design 100,000 things on a DNA chip. So maybe I'm like, I think just mass is good. Diversity, kind of new topologies is good. I think, interestingly, a lot of the real research push right now has been on kind of monomers, new topologies, and I think there's good stuff to do there. It's like unclear, especially let's say for kind of tricky interactions like peptide and antibody interfaces. Like how much does like what you learn about what packs a monomer inform you about kind of the physics of like weaker interactions? So that's another one. It's kind of good protein interaction data.
I'm going to sneak a question in. So I think a little bit after this paper came out, RF diffusion came out, and it was quite a contrast in reading. So yours was this sort of like super kind of first principles derivation from very clean mathematical rigorous, and then there's somewhere in there, but it doesn't like read. It reads very differently, and they kind of repurpose AlphaFold, and it's like kind of, I would say that's an engineering paper, and yours is like kind of more of a math paper, and yet you're both doing essentially trying to solve the exact same problem. And I'm just curious, like, what your thoughts are on like comparing and contrasting either the development or the outcome of those two things. Or anything, any comment at all on the differences?
Yeah, I mean, I think they're both very good works, and I think like there's a certain degree of pragmatism you have to have with all of this. So I think, for example, with that, like if we tilt towards like what structure predictors know, self-consistency of structure prediction. I mean, I think what they did an awesome job showing was like we can actually reduce to practice a molecule that does this thing, like experimental validation of a lot of things. With this one, I think with what we're putting out into the world, at least, like we were more kind of, it was a little bit more academic. Like, what if we sample from the spicy distribution, and we're kind of not trying to like get the thing that will land like the great this will work figure. And we're also kind of just interested in like, can we get metrics out of these? So we tried to be very like likelihood based. I think technically like our schedule that. One thing that I think is kind of interesting, this happens in image world too, where everyone likes like FID scores. And then it's like the few papers that are like, oh, but like what's held out likelihood? I think right now the backbone modeling world is like, what's likelihood? Or there's like no notion of kind of validation set generalization. So we also try to look at those things, but I think now just the question is kind of just how do we keep doing good engineering to build models that can leverage data fully. So
maybe just a quick specific follow-up question. Like you have this non-IID, you have like a non-diagonal covariance matrix with this collapsed polymer prior, and then you have this sort of like sparsity, and like I think they don't have any of those things. Or am I mistaken?
No, they do. They don't have those things, but they also have just like the hardcore full structure prediction, all interaction, which can work well. So I mean,
like, so if you were to go back, like, so if you wanted to bet your money on like the quickest path to like making proteins that are useful and work, and you were starting from scratch, like, would you go the clean academic route, or would you like repurpose AlphaFold into a diffusion?
Oh, I mean, I'd do what we did. Okay. I think, we're also, a lot of the stuff we do, we don't like, let's say, talk about in this paper, but where we do a lot of antibodies, and they're really at the edge of physics. And so at least in kind of our hands, it seems just like thinking about kind of where robustness comes from, what do likelihoods mean, has been helpful.
Okay, thank you.
I have a pretty basic question because I don't really understand diffusion models. So we saw that in the problem of mapping from protein sequence to structure, like the models, current models are not very good at understanding the impact of mutations, understanding like things that we haven't seen in nature, what they would look like. And it seems like you can use diffusion model to design something completely new and get reasonable structure. So can you explain in very plain language, like intuitively, how these models learn the things that these structure prediction models are not able to learn?
I don't know that I would kind of view it as like there's something structure prediction models can learn and something that like these can't. I think it's like more kind of just this classic question of like factorization and variation in data. Like if I have, so one version of it is if I have a large corpus of data that has like some degrees of variation that are very undersampled, and then some degrees of variation that are very sampled. So I would say maybe proteins are this. Proteins on Earth are this. We had like a small number of folds that mostly got invented several billion years ago. And then we have like the metagenomic mega sampling of kind of solutions to those folds. Sometimes that's like a thing where kind of what you need to do to generalize in the fold problem is very different than what you need to do in the kind of the conditional sequence given structure problem. So I think it's more just like these types of factorizations maybe allow us to build weaker models that have no chance of being good at structure prediction, because they're not like learning the kind of the detailed family specific statistics of kind of like proteins on Earth. But because we can make them so simple, they can kind of generalize across these like coarser classes like folds in ways that things that are trying to model, let's say like kind of all of the implications of like sequences on Earth are going to have a harder time doing. I mean, it's all, the distillation stuff's interesting. There's kind of probably, we'll keep seeing like blurring of all these things.
I think we have time for one more question, but I'm going to first sneak in. There's really a strong request for no food or drink in the auditorium. So, okay, but now then we'll go for lunch.
Yeah, thank you really for this really nice presentation, both technically very deep and also very consequential. Actually, I had like one small observation, and maybe correct me if I'm wrong. So at the point where your generative process becomes sort of a multivariate Gaussian with off-diagonal coupling, right? So it's basically like a Gaussian process, right? You sample from a multivariate Gaussian. Shouldn't then also the noising process be sort of a Gaussian process, so that you basically also have couplings in the noising process to kind of make the posterior variational distribution match better? Because ultimately you derive these sort of Gaussian variances systematically from an evidence lower bound, right? If you derive the diffusion model. So I could imagine that you might actually get a tighter bound if you also make the forward process sort of multivariate. What would it basically mean is that you know, when you create noise, you're basically creating correlated noise, right? You kind of still have, you make sure that in the noising process, like nearby bonds are still preserved, right?
So I may need to talk to you more to like clarify I'm understanding. So we do have, our forwards process is correlated Gaussian noise.
Right, but it could be correlated noise across pixels, right? And I'm just wondering if that...
So it is.
Oh, it is already, okay.
Right. So it's like kind of the full, yes.
So both forward process and backward process are sort of correlated.
Well, the backwards process is still kind of using the standard denoising diffusion, which I think might have, I mean, that's interesting if like you take the VAE perspective as like the implied
so I think you could make both of them multivariate, but yeah, we can chat offline.
That's interesting. Yeah.
All right, cool.
All right, I think we gotta head out to lunch, so we'll be back. Today the next talk is not at 2 like yesterday, but is at 1:30, so don't forget. Yeah.